time. So first, again, I'm Linda D. from DARE, Community Engagement Coordinator, joined by Michael Llewellyn, the CAB co-chair. And first, let me introduce our two speakers who I know very well. First, Dr. Kamel Khalili, who is Dr. Crispr, I think would be appropriate. He's got more than 35 years experience in studying human viruses, including HIV-1. He's received his doctoral degree from the Department of Microbiology at the University of Pennsylvania and spent 3 years as a Fogarty Scholar at the NCI, which is, of course, at the NIH, where he collaborated with several scientists investigating HIV-1 at the early stage of the HIV pandemic. He returned to Philadelphia in '87 and continued his research on HIV-1 and other human viruses. In 2013, he developed a strategy for eliminating HIV-1 DNA from the host genome using a gene editing technique called Crispr. Therefore, Dr. Crispr, and his goal is to utilize this novel approach for a cure for HIV-1. He founded several departments and centers at 3 different universities in Philadelphia and is currently the chair of the Department of Microbiology, Immunology, and Inflammation, and director of the Center for Neurobiology and Gene Editing at the Katz School of Medicine at Temple University. All right, let me say that ATAC, the AIDS Treatment Activist Coalition, met with Kamel. We were trying to figure out the amount of years. He said 5, 6, 7. I think it's more like 10 years ago when he first presented this technology to us. Anyway, also joined by Trish Burdo, who is a professor and vice chair in the Department of Microbiology, Immunology, and Inflammation, Center for Neurobiology and Gene Editing at the Lewis Katz School of Medicine. Trish is an internationally recognized immunologist, has made seminal contributions to the role of chronic immune activation in HIV comorbidities, which we were also talking about at the beginning of this, earlier today. The Burdo Laboratory led key studies on the relationship between monocyte activation and myocardial pathology and neuro-HIV in the non-human primate SIV model. The laboratory also demonstrated SIV proviral excision by CRISPR-Cas9 editing in the non-human primate model. Dr. Burdo is one of the directors of the Crispr for Cure Martin Delaney Collaboratory, director of the T32 Training Grant, and co-director of the Comprehensive Neuro-HIV Center. She has served as the HIV Comorbidities and Clinical Studies section, and the NIH Office of AIDS Research as a member of the OAR Advisory Committee, which is where I met her when we were both on the ORAC at the same time. So welcome to you both. And as I said to Trish, what a mouthful, very eminent researchers. Go for it. We appreciate you being here, and thank you very much for taking
thank you very much. Thank you very much, Linda, for kind invitation. On behalf of Team Crispr for Cure and my colleague and good friend Trish, I'd like to express our gratitude for the opportunity that you gave us to share some of our efforts that we've been doing to eliminate the virus agent that we have been working, as you mentioned, for more than 40 years, since the earliest days of the pandemic. And knowing that the virus has been a very big challenge for in health, and in particular with more than 60 million people, as you all know, got affected. Half of them unfortunately died. The other half are still in struggle of how to get rid of the virus. So let me start first with with my disclosure that as you see that I'm on the board member and scientific advisor and hold equity on the company by name of Excision Biotherapeutics, which is a startup company for licensing of technology that we develop at Temple for HIV Cure using a gene editing approach. So Our team is rather small, but highly interactive and spread across the country, United States. And we have been working with each other for many years. And while it's small, but it's highly interactive, and we have been publishing, we have been focusing on the way of getting rid of the virus for different approach. And the Crispr brought us together and then developed the program. And you see that in this slide illustrates the institution who are involved in this program. So, of course, community engagement plays a critical role in what scientific strategy that we are using and our progress. And Sophie from PEP is the leader of this community engagement, who was led by the Vanessa Perron, and also Kate Goldstein is a coordinator of our program. So our program has basically 3 focus area, and then and as you see here, many of you probably know these are the distinguished, highly accomplished HIV scientists. Well, we were very fortunate to have them to work with us. John Carn is a colleague and friend for many years with me, and also Jerry Sack, that the very known accomplished HIV scientist. And they both lead the Focus Research 1. And other co-investigators are also highlighted in this slide, as you see it, including Trish and Shane, Brian Bigdahl, and Mark Nanomarker. And then on my next slide is research, or we jump to the research focus 3.
I can go back. There you go.
Research focus 2. This component of the program is mostly involved in the preclinical studies that Trish is in the non-human primate, and Leon is on the new humanized mice that he developed in his laboratory. Very unique and very useful to understand the pathogenesis of the disease, and also useful for elimination of the virus using the gene editing strategy. So the research focus 3 is basically focusing mostly on the gene editing and delivery of the Crispr in the cell culture and animal model. In this program, we have Hairun Cho and then Mike Farzan, who are expert in AAV for delivery of the therapeutic molecule, are involved in the modifying the technology that they have and make it the T-cell trophic and or macrophage and the myeloid cell trophic, and then Brian Wigdahl and then TJ Craddock and Marker are involved in the ensuring that the technology that we have are safe and it does not cause any off-target effect on the host cell. So basically, they're involved in helping us in developing the most efficient guide RNAs for elimination and editing of the HIV without any impact on the host cells. So now, yes, please. And we also have a corporate partner, which is Excision Biotherapeutic, which is headed by our CEO, Dan Dornbush, and Jen Gordon. Jen Gordon used to be a member of our team, but she moved to California, and right now she's the vice president for clinical R&D of the company. And also we have Mark Lewis, who is head of the ICOL, who's handling a lot of non-human primate studies that we are doing, and work very closely with Trish, and in the past also working closely with myself. It's a terrific operation in the Maryland, Rockville, Maryland, that is very helpful and essential actually for a lot of work that we are planning to do through this Martin Delaney program. But let me just go a little bit about the science without getting into the detail. This slide actually illustrates that what you see in the center is a cartoon of the cell that gets infected with HIV. Just assume that this is a T cell, and then T cell is a membrane which is decorated by the group of protein who are serving as a receptor for the virus HIV. And once the connection between the receptor and those spikes protein on the membrane of the virus takes place, then the virus gets into the cells, enter into the cell, and viral genome, which is two copies of identical RNA, enters into the cell along with the enzyme reverse transcriptase. So reverse transcriptase is basically enzyme which converted the RNA molecule to become a DNA. And then that DNA finds its way getting into the cell and integrates in the host genome. That's the stage that the virus remains either at the silent or can be activated by the host proteins to start producing the new progenies. That new progenies are basically from the virion and budding out from the cells. So if you see in the side, there's a picture behind that.
Oh, maybe that's over.
Yes.
One more.
Maybe. No,
Oh,
maybe there we had the picture showing that exactly in real time that the virus is budding out from the cells. But anyway, and then the newly synthesized virion can get infected the other cells, and then life goes on for the virus, but the cells can get killed. What the antiretrovirus has done so far successfully is either or block the process at the earlier stage of the integration of the viral DNA to host genome, or after the process of the viral RNA replication processing of the protein. And the result is that the virus cannot be productively infect and replicate in the cells. So one thing that the antiretrovirus has not been able to do that, and actually it's not programmed for that, is that the affecting integrated virus in the host genome. So in another word, then when the patients or individual are walking in the AIDS clinic, they already have the virus incorporated in their genome. So what they can be done for those individuals is just put them on the antiretrovirus. So basically block the viral replication, restore restore the cell counts, and then inhibit the viral rebounds. But the viral DNA remains almost intact, and then the majority of them some of them are mutated, non-replicated competent, and some of them are replication competent. And then anytime that the antiretrovirus is stopped, then those replication competent virus can replicate in the cells and put the patients at risk for developing of AIDS. So this is basically schematized the chromatin and then the DNA healthy part of the cellular gene, which a viral genome is integrated. So the strategy that we used and that was basically many years ago, was tried to permanently inactivate the virus genome in a way that even if there is no antiretrovirus, the virus cannot basically replicate. To do so, we thought probably if we can find a way of cleaving a viral DNA from exactly the regions of the integrated without any damaging, any unwanted editing of some other area of the cellular gene, that may lead to the permanent cure of the disease, to the point that we no longer need to get any antiretrovirus because there is no virus to replicate. So our strategy for doing that was
I got it.
You got it. Our strategy basically was to cleave, as you see here, two ends of the virus, or at least a part of the virus, make a replication competent virus to become replication incompetent. So as a result, then the virus become permanently inactive. So so I have a little bit a minute or so clip that shows that how the CRISPR-Cas9 technology can work.
There are two components to the system, a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes. There are two components to the system.
So the strategy that we used to employ the CRISPR technology was first we started in 2013 and employed to make sure that the CRISPR is able to eliminate the virus in the cell culture system. There are a number of cell models for HIV infection and with T cells and also macrophage. So we use that one and we showed that for first time we were able to demonstrate that you can actually eliminate the virus DNA from the cells without any impact on the host. And basically the cell became completely normal, maybe right before like a cell without any viral infection. So we extended that observation that tried to see that we can replicate that the cell culture in the patient samples. So we were able to also achieve that goal. So obviously the patient samples came to the laboratory, we cultured, and we applied the CRISPR technology, and we showed that the virus was eliminated from the cells. Then we had to move it to the in vivo model. First, we started with the humanized mice, a small animal model. So in this mice, basically, we programmed the animal with the human cells, which is susceptible for infection by HIV. And then after that, we introduce CRISPR by IV inoculation, tail vein inoculation of the CRISPR using AAV9 as a delivery. And then after the several weeks, while the virus was replicating in the human cells, in the body of the small animal, we apply the CRISPR technology and see whether or not the development of or presence of the AAV9 CRISPR can lead to the suppression and elimination of the virus. So, then based on that observation that we have, next step for us was using in vivo non-human primate that Trish is gonna a little bit elaborate on that. And that was important for the safety and efficacy of the technology. So this is just one slide of the study in humanized mice that paper was published and earlier, and then that again for first time shows that you can actually use a CRISPR technology and then eliminate the virus in the humanized mice. And then the top panels shows that basically those dots that you see are representing the virus or viral RNA, and it showed that when you have the CRISPR applied to the animal whose virus is controlled by ART, result in the complete elimination of the viral DNA. And actually, biochemical assay also show the absence of any evidence for the viral genome, and in these two particular cases. So the rate of success at that time that we received, we reproduced this experiment 3 times. In one case, 2 out of 7 animals. The second one, we have basically 5 out of 10 animals, and then we reached to the higher level eventually by manipulating a little bit the time of application, and then when applying and the dose of the virus and a few other adjustments. So, but the outcome of this experiment was that it's showing that you can actually eliminate the virus in vivo, HIV in vivo by CRISPR technology. So one experiment that also gave us a confidence was that we took the spleen, bone marrow, and lung from the infected animal, which showed the rebounds because of the lack of the CRISPR, and injected in the uninfected mice. And then after 4 weeks, we look at the spleen and the bone marrow and look for the viral DNA. An assumption was that if there is any viruses present still in the infected animal after rebound, it will be able to detect it in the uninfected animal. And the outcome of that shows that the animal who received the rebound due to the CRISPR technology shows basically no presence of the virus in the secondary infection. And this is very powerful adaptive transfer technology, which basically gold standard illustrating the complete elimination of the virus in the by CRISPR in the initial infection. So now having that study in the animal, now we are basically did a lot of safety and efficacy experiment in the in vivo that Trish is going to mention, and based on this study that we received from the non-human primate and also the humanized mice, we developed IND, so applied for the FDA and received approval for entering into the clinical trial. And then the clinical trial actually started about the limited, small scope clinical trial started by Excision Biotherapeutics that is basically handled by the CEO of the company who have developed the clinical teams for performing the clinical studies in the number of the sites. One of them is in St. Louis, and the other one, a couple of them are going to initiate in different part of the nation, including one in the Camden Cooper Hospital and also in California. So with that note, I will stop and let Dr. Burdo present some of the observation that she has on this project.
And while Kamel was talking, I have been looking in the chat, so we will cover some of this stuff. And I think that if we don't cover stuff, then we can come back to it. And I apologize, my lip is bleeding. So I don't know what's happening, but so I will start my part. So basically the first thing I wanted to do was kind of break down some of this really complicated stuff that Kamel was talking about. And really I wanted to focus first because this talks about some of the off-target things that people had asked about before. So the first thing really is that HIV DNA gets into the host DNA of the cell. So Kamel went through some of the viral life cycle, but really the important step for the CRISPR here is that HIV DNA can get into the host DNA. And so this is when people are on antiretroviral therapy, it blocks every step pretty much if you're on, depending on what antiretroviral therapy you're on, except for the DNA. It never removes this DNA. So that's why when someone stops their antiretroviral therapy, the whole process can start over again because there's DNA there. And then the ART does not get rid of the DNA. So, we talked a lot about gene editing, but really I wanted to say, what is gene editing? So this is a technology that allows genetic material. So genetic material includes, DNA to be added, removed, or altered. And this can be changed or swapped out at specific locations of the genome. So what we're not adding anything, we're not altering anything. What we're doing in our strategy is simply cutting out HIV DNA from the host genome. So as you can see here in our little, let's minimize you guys. As you can see here, when HIV incorporates into the host genome, so this is DNA, and the purple here is the HIV DNA. What we're doing for the strategy is using gene editing to make a double-stranded break cut here, like the video showed, and here. And we're doing that in a number of different ways. But essentially, what happens is then a large chunk of the virus is removed. So we want this to happen. We want this to be double-stranded break, double-stranded break. And then what happens is the body has this innate ability to repair the DNA. And so then the DNA here is repaired. So that's essentially the strategy that we're using. And it's different from other strategies for other diseases and for other people are using different kind of CRISPR. So essentially then what happens is, oops, my X went over a little bit. But the HIV is removed from the host DNA, and it can no longer make HIV after you remove ART. So this is the basic strategy, is that you remove the DNA. So then if a person after that, they remove the antiretroviral therapy, this is how we're trying to, do HIV Cure. So I'm sure, Kamel showed you a really cool video of CRISPR, and we've all hopefully heard this word CRISPR and Cas9 and all these things. But so CRISPR stands for clustered, regularly interspaced, short palindromic repeats. It's a big mouthful. But essentially what CRISPR is it's a gene editing tool that has two components. The first component is called the Cas9. And this is just CRISPR associated protein. So the important thing about this protein is this protein clips the DNA. So it makes that cut on the DNA. And then the other component is what we call a guide RNA. So the guide RNA identifies the gene of interest. So it really tells the Cas9 where to cut, right? So you want something that cuts, but you want something that says where exactly to cut. Right, because you don't want it cutting DNA it's not supposed to cut. So CRISPR cuts DNA at a specified location based on this guide RNA. So it guides it there. And this gets into some of the off-target things that I'll discuss in a little bit. The other thing that I think is important to look at our strategy is we're looking at an in vivo strategy. So I'll explain this. So there's two different methods in gene editing. One is ex vivo, that you take cells from outside the body, then you do the gene editing, then you would put those cells back in. Right, so that's here on this side. This is, removing blood cells from patients, doing a transformation with the viral vector, expanding them in culture, then reinfusing them. And this can only happen in blood compartment. You can't take out tissues and put them back in. So this is only blood. And then also, this needs a lot of equipment. So you would have to remove the blood, manipulate it outside the body, then put it back in, and also takes more time. So the strategy that we're trying to develop is called an in vivo method. So you would directly infuse, by slow infusion, this the viral vector with the Crispr in there. So we think that this is more scalable, easy to apply, and it gets into a broad range of tissues, and it reaches lots of tissues. So from our monkey studies, we see that it can reach a lot of tissues, including brain, which is important viral reservoir. So where the virus is hiding in the body, it can reach the lymph nodes, it can reach the gut, all these different things. So we think this is gets to more tissues than the ex vivo portion. So delivery. So I know there's some questions about vector delivery, and I'll talk about that. So our initial strategy that we're using is to use adeno-associated viral vector 9. So this is going to be used to transport the Crispr inside the body. It's similar to vaccine strategy in a way for the delivery. So AAV9 will then include this DNA, which then makes the Cas9 and the gRNAs. So that will go into the cell, and it will just release the CRISPR Cas9. So that, again, the Cas9 and the gRNA. And then what happens is then it can then go do its job, and it can cut the DNA. So AAV vectors have been used in a lot of clinical trials, and they remain a key delivery for this CRISPR system because they're highly efficient. So they have a high efficient delivery. So what are some of the advantages of using this system? So this system can be precise. So it's precise cutting. So it only will cut the HIV DNA. We think this will be a permanent removal because once you cut it out, it doesn't go back in. It's a promising genetic tool used in HIV, but other diseases, and they've shown positive results with this and things like hemophilia. And also another thing is the edited genes are not passed on to the offspring. So this, there's no worry there of whatever, the editing happens will be passed on. And third, our non-human primate studies have shown us that CRISPR can reach multiple tissues, which we call viral reservoirs, and then cut out the DNA. So to get to, I think, a lot of the questions that people have is, how are we measuring safety? Is this safe? So the first thing is that we want to make sure that these gRNAs can cut only HIV and not the human genome. So this, this means it's very specific. So we do a lot of designing from the beginning before it even gets into an animal, before it gets into a mouse, before it gets into any system, to make sure that gRNA, like the guide RNA, cannot, it doesn't match with any human DNA. So that's one thing. The other thing we do is, we're using the animal models to test the safety. We look at liver and kidney function. We look at tissues to make sure that there's no pathology there. We make sure, we look at the animals to make sure that they have healthy blood counts, that there's no inflammation. And again, I saw a lot of questions about AAV9 and antibodies against the AAVs. So, there is antibodies against AAV9. Not all of them are neutralizing. We have not found in our studies that this affects the efficiency of the CRISPR Cas9 delivery. But we are aware, there are a number of other AAVs that could possibly be used. There's AAV9, but there's AAV6 that we've used in studies. Some of those antibodies do not interact with each other. So there's definitely other ways to do it. Someone asked, what if you need more than one shot? Again, this could be a way around that, is to use different AAVs. Another is, someone asked about different delivery systems. So there's lots and lots of studies going on about different deliveries. So nanoparticles, which are basically lipids, so fat that you can put this into and get it into cells. So if anyone had the COVID vaccine from Moderna or Pfizer, basically that's a lipid particle. So it'd be something similar to that. So there's lots of different opportunities. Also, we look for extensive off-target analysis. So we look for unintended genetic modifications. And so we have done extensive studies in our monkeys and our mice to look at a number of different genes, number of different tissues. And the good thing about this is, since it's a non-human primate, at the end, we do a necropsy and we can look at every tissue and do a lot of sequencing, and we put a lot of money into really looking at this. And to date, we haven't found any off targets. And I think this is, not unexpected, because we're cutting out a viral DNA. Some of the other strategies for genetic modifications are looking at, human genes. So we're not looking at a human gene. This viral DNA should not be there in the first place. It's not a human DNA. So we're removing it. So hopefully that answers some of the questions so far, but I can take, I see one that says, are we aware of any tissues or sites that are not reached by CRISPR? So I think that, I think one of the big things is finding the correct delivery, but also the correct amount, right? So you can give different amounts of AAV. I think that's one thing. So we're still looking at increasing the amounts that you can deliver with still being safe. So that, I think that's one thing. So someone asked, will this prevent a potential future infection? So we're not using this as a prophylactic. It's not similar to PrEP, right? So we're removing the DNA. So I think that, this is really a cure. But I think in the case, if I'm not correct, even in the case of people who were cured by bone marrow transplantation, in those couple of cases, I think, some of them would choose to go on PrEP, right? So then I probably would think it would be a similar strategy once, so you're not reinfected.
Yeah, can I add something? Yeah. Actually, it can be used for prophylactic, but you have to manipulate the expression of the Cas9. We did publish a paper in about 3-4 years ago and put the Cas9 expression production under the control of the viral protein. So in other words, that every time that the cell gets infected with the virus, viral protein stimulate expression of the Cas9, and Cas9 is targeted for the cleavage of the viral DNA because of the guide RNA, as Trish mentioned. So immediately gets eliminated without any chance of integrating into the host genome. So the whole idea here is that to prevent integration of the viral genome in the host gene and eliminate the replication competent virus, which is already integrated in the host gene. So there won't be any need for the use of the antiretroviral treatments. So yes, it can be used for the anti-prophylactic approach, but you have to plan for it. It's different than what we described.
Sure.
Can I ask you a question now, Trish? There are a couple of questions, and I think you talked about the off-target issue, and sure. I'm wondering what do you do to study that continually to make sure that's not happening? And how about new mutations over generations? I mean, is that an issue? And also, finally, the AAV-based treatment safety issues that we've been hearing about. How do we get around that?
Yeah, so that's a pretty loaded question. So, so I'll take one part at a time. So the second one where you said the generation-based mutations, you mean per individual or virus mutations?
Individuals.
Sure.
Okay. Yeah. So we are designing these guide RNAs, so they're pretty universal, and so we've done a lot of sequencing that shows that they're appropriate for a number of over 99% of the viruses, and we're looking at really conserved regions, and we're also not just using one target. We're targeting in the virus, we're targeting the LTR, but we're also targeting GAG and the other LTR. So we're targeting multiple places too. So we have a better chance of removing it with these multiple targets. Go ahead, Kamel.
I think, Linda, your point is that whether or not there's mutation occurs in the patient, individual, later on in life, and whether or not the CRISPR can cleave that cells. But remember that the CRISPR that we're applying is not going to be there forever in the patient. So usually after about 2 or 3 months, basically there is a viral DNA, AAV9 is not going to be there, and then it won't express CRISPR-Cas9. So if there is any mutation later on occur in the host gene, which is compatible with the guide RNA, may not be as effective because there won't be any guide RNA, or there won't be any Cas9 even in the individuals. The important here is that at the time that the treatment is initiating, there won't be any chance for the off-target effect. So after the application and elimination of the virus, after maybe a year or 2 years later on, if there is a mutation happens in the person, which usually happens in the individual because of let's say that some part of the gene gets mutated because of the repair pathway in the person is not correct, then at that time it doesn't mean that the CRISPR is still exists to cleave the DNA. I don't know if I answered that question or not.
Well, it's very helpful. And what about the AAV-based treatment? I mean, some really serious safety issues have, we've seen in a couple of papers. Can you get around that by using? I don't know. I'm not really sure what these different numbers of AAVs mean. Or some less.
No, Candelaria. Sorry, CAN. Hey, how are you? DE.
Everybody that's not speaking, mute, please. So anyway, is there a way to get around the AAV issue?
Well, so far AAV9 has not shown any big impact on the health of the individual receiving AAV. AAV9 right now in the clinic in the 8 or 9 different clinical trials, and there has not shown any adverse effect. So there are some sporadic paper comes out by some of the people about the different kind of AAV, which was used for infant or at the extremely high amount of the AAV. So obviously the safety of that one is some issue.
So I think that's one of the big things. I think it's the dose of AAV they're putting in, and so I think that's one thing. Some of the things that have shown these liver toxicities and stuff were something that was it was a really high dose, and also it was infused in a small volume. Instead of doing, so we do a larger volume, and it's like a IV drip. So it'd be dripping over time instead of just like, because if you just squirt it all in there, then it goes right to the liver. And I think that's some of the issues where it was a lot of virus, and it was a really, concentrated amount in a short amount of time. And also some of the ones have been there's different things where they use different serotypes like AAV8 or AAV1, but AAV9 has been used in a lot of trials and haven't had that same type of issues as some of the other ones.
Great, thank you.
Mm-hmm. So I also see a question about what if we don't get the entire virus, and could things like TAT and VPR, still be produced? So the strategy we're using is the best case scenario is an LTR to LTR full excision, but also the possibility of LTR to GAG and GAG to LTR. So a lot of those, the DNA will be eliminated in those cases. And so, there
won't be any TAT.
Yeah, there shouldn't be any TAT. We're also working on additional strategies to, that are TAT specific strategies within the Delaney. So we would have to make it cell specific. If TAT's being expressed, then the Crispr would be turned on and then cleave those cells too. So we're trying to incorporate those things as well into our strategies.
Great. So I think that a lot of the questions have been covered. Do people want to jump in with any questions? Jeff Berry, we have Jeff Berry, the co-chair of the Crispr for Cure CAB with us. Is there anything you wanted to add, Jeff?
No, I don't think so. Thanks, Trish and Kamel, for such a clear presentation. I think Maria had a question, and then I just want to make sure Corinne's question at the beginning was answered to her satisfaction about off-target viral cutting.
Yeah, Siggy too. Siggy, Corinne or Siggy, if you want to jump in with those kind of questions, now's the time, and then we'll do Maria.
Yes, I put my question already in the chat. By nature, double-strand breaks cause error-prone repair. And since you never know what is introduced into the genome by this error-prone repair, it could be extra nucleosides, it could be frame shift mutations, it could be stop codons, whatever. But you cannot predict all possible scenarios. So even if you haven't seen any unwanted effects so far, that doesn't mean there won't be any at all. And that's a principal problem of this kind of approach.
Well, yes and no. As CRISPR cleaves, as you know, that there are a number of DNA repair pathway in the cells. One of them is homologous recombination, and the other one is non-homologous end joining. And non-homologous end joining is the one who basically joins brings together the two ends after cleavage by CRISPR. And that is subject for introducing indel mutations. So the strategy that we have here is just cutting two ends of the viral DNA. So when the non-homologous end joining brings this DNA back together, and even if there's a mutation happens, it's not bad, because after all, those mutation makes the virus inactivated. But
but the virus is inserted in a gene, and something happens with these genes after the error-prone repair.
That's what we have to make sure, that the off-target doesn't happen. And for that reason, we didn't go through the...
I'm not talking about off-target. I'm talking about the error-prone repair mechanism that introduce mutation into the gene where HIV used to be.
So we have done extensive ultra deep sequencing of the after the treatment with the CRISPR, and we have not seen any such a mutation after the elimination of the virus. And in fact, all the data that we are mentioning regarding off-target was submitted to FDA during the preparation of IND. So
yeah, and if you think about it in the opposite way, so what happens to a gene once HIV is inserted, the DNA is inserted or integrated to there, right? So we're not introducing anything additional, we're removing something born that wasn't there. So if there's any changes in that gene originally, it's probably from the original integration.
Yes, but that probably means that the function of this gene is just disrupted. But once you remove HIV and introduce some other mutation, it may mean that the gene product is produced again, but it's not the original gene product, but it is a slightly altered project or completely altered in the kind of a frameshift mutation.
Yes, frameshift mutation, it means that the function of that cellular gene is no longer the same.
Exactly. And that may or may not cause problems.
Yes, but the thing here is that that gene was non-functional because of the viral integration. So
yes, but there's a difference between a non-functioning gene and a wrong functioning gene.
So you're talking about the gain of function as a result of the excision of the
For example.
Well, this is something that we have not seen. It doesn't mean it doesn't happen, but
that's exactly my point. Thank you.
But you have to actually at some point look at the benefit of the elimination of the viral DNA from the gene for a very rare event that may or may not happen. So
I
totally agree.
You're watching for that is, I guess, the question. Are you haven't seen any signals? Are you watching for such signals or?
Oh, absolutely.
Yes.
Yeah, we're looking at other things like AAV integration. We're looking for, any of these frameshift mutations. We're looking for any chromosomal alterations. So we do a whole extensive off-target portfolio. And that's one of the things, we've been doing in the animal model and cell culture, and it will be done in all of the clinical trials once they're underway for the company.
So I think we need to probably move on. Let's go to Maria and then Mark Wagner and then Nelson. We need to make it short though, because we've only got a little bit of time left, about 5 minutes. Yes. So Maria, you want to go?
Yes. Mm-hmm. I really appreciate it. I want to say, first of all, quickly, thank you so much for all the work that you're doing. All you researchers, physicians, Dr. Kamel.
Yes.
Just everyone. Thank you very much for even letting me be in this space. I'm just a long-term survivor of 34 years, and I do have hope about the cure. And I'm just going to pass on this information to all the following that I have globally. My question is very quick and simple, and excuse my ignorance, I'm not a physician or a scientist. The late Timothy Brown, my very, very good friend that passed away, unfortunately, of cancer in 2020. We all know that the man bone marrow transplant, there unknowingly, he got the mutation that eventually eradicated HIV and made him HIV negative again. So I always thought to myself, and I've always asked myself that question, I believe the cure is in genetics. Why can't you emulate in a lab that genetic mutation and inject it in us? You know what I mean? I don't know if I'm making myself clear, but that specific genetic mutation, why can't you put it inside our bodies? I don't care if the virus is there, as long as I don't have to have pill burden. I'm tired of it. So is this something similar to it? Because it's genetics. Thank you.
Where are you from, Maria?
Fort Lauderdale.
I was born in Medellin, Colombia, but I was raised in Miami and currently live in Fort Lauderdale, Florida.
Oh, great. Activist. Okay, thank you.
Yeah.
So we can answer that for sure. So we're working on those strategies actually, and so we're working on the CCR5 CRISPR strategy. But there's some questions raised here about, how are you going to not interrupt some of the host genes and other hosts? So when you're using a type of strategy like removing the CCR5, which is essentially, what happened with Timothy, there's something else happening because you're now targeting a host gene. So, human genes are more like human genes, or viral genes different, right? So I think we're working on those strategies, but it's not as simple as it seems in reality, right? So, and I think, safety is a big issue there, and also longevity. So how long is that going to last? A bone marrow transplantation, you got rid of all of the cells and put all new cells in, right? But T cells and stuff, those things turn over. So is that mutation going to stay with, or is it going to come back, right? So it's a different strategy, because you would have to make sure you remove it all, and especially from the bone marrow, right? You would have to. So it's a different, more complicated strategy, but we're still working on that aspect of it as well. But we think this is targeting the HIV itself is a more direct strategy. I think.
Great. Okay, I'm just going to go down on alphabet. Oh yeah, go ahead. That was it. Mark, Nelson, and Khalili. And then we'll have to cut it off after that because we're at time.
Right.
Mark, you go.
Mark, you can unmute yourself.
Did you want me?
I don't know. Mark Guest.
Okay, you can do.
And Mark Milano had my hand up too. I don't know which one.
Yeah, Mark Guest.
Okay.
That's you, Milano, I guess.
Yeah, go, Mark Milano.
Okay, all right. Sorry. So I'm an HIV educator. So my question is basically for all my clients, timeline. What's going to happen? What's the next step? Is it in humans yet? When will it go into humans? What's your hopeful timeline for moving this into serious human trials?
Well, right now it's started in a human, and obviously under the close monitoring on the viral load, and see that whether or not the virus is coming back, and if it's coming back, how is it compared to the control? Is it any delay in the viral rebound or not? So these are the questions that needs to be addressed clinically. And then, of course, the individuals are very closely monitored by the cast of the clinicians. And all those studies are done by the Crispr, by Excision Biotherapeutic team. So we are not basically too much involved on that process. So I cannot answer precisely your question. But you can certainly contact by email, send me email. I can pass it to Excision Bio team so they can get back to you.
Yeah, so Linda, we can send you the contact at Excision Biotherapeutics, and as Kamel said,
yeah, I have it. I have it.
Yeah, our CRISPR for Cure Martin Delaney, basically does not do clinical trials. We're on the basic science part, so we're trying to work on making this as safe as possible, as specific as possible, by using a lot of mice and animal models and monkey models. So that's kind of, and doing all these things, improving, like you said, improving delivery, improving the editing, improving. So that's where we are.
Yeah, they're doing a trial now, Mark. So that I know of. And then we have, let's go to
Nelson.
Next, Nelson.
Yeah, I have a very general question. When it comes to data from human exposure of Crispr in other pathologies, I've seen some studies on different blood cancers and all that. Do we have an idea how many humans have actually in different pathology studies have been exposed to CRISPR in the public domain, that we can actually, feel a little confidence that we're not the first? Obviously, HIV is not the first cohort, right, that is going to be exposed to the CRISPR technology.
I personally don't know that, but we can definitely do some work on clinical trials and stuff and try to figure that out. But yeah, that's a very good question, and I don't think I can give an exact answer.
Thank you.
And then Michael, you know what? I always forget this. Could you put the evaluation in the chat? But you need to change the date on it in the title.
I don't have that, but I'll try to get something up there. That was Richard who was doing it the last time. I don't know if he still has it.
I think I put it in last time. I gave it to you last time, and It's a new one but it's, it's got the wrong title on it it's because it's from last time.
Right. Bahati.
Hello.
Hello.
So I have two questions. I'm just trying to understand all that has been presented. My first questions, I want to know, what side effects are we to expect if this trial is done in human beings? As we know, it's not a natural removal of the HIV virus. So what side effects are we to expect once this is done in human beings? But also, I wanted to know on the measuring safety on the first point, is there a percentage of which we can measure the efficiency of the gRNA in cutting only the HIV DNA and not any other part of the DNA? Is there an efficiency level on that? Thank you.
Let's take the second one first.
Yeah. Hopefully, we won't have any side effect, adverse side effect in the application of the CRISPR. And actually, we are in process of publishing a paper that Trish basically led for that paper publications. Most of the work is done by under her supervision, and we actually, surprisingly, with a pleasant surprise, we didn't see any adverse side effect in the non-human primate. In fact, some of the animals start gaining weight, and there was a improvement in the virus the cells, lymphoid cells, and it seems that the elimination of the virus or decrease in the concentration of the virus in the non-human primate is beneficial for the health of the
pathology,
for the pathological studies also. Yes.
Sure, and so we don't want to say that there's not going to be any side effects, because of course, if you get anesthesia, or if you get a needle, or if you get some type of infusion, there's, with all of those, no matter what they're infusing into you, there's always a chance of side effects, right? So there's always a chance of some infusion reaction. There's always a chance of getting redness and soreness and all those things. So I think our point is that we haven't seen anything that is specific to the AAV CRISPR in our studies yet. But it doesn't mean that it's going to be free from side effects. I
think that's a clinical trial. So clinical trial is going to teach us something that we don't know in the effect of the techniques and approaches in the clinic.
Yeah, and someone just said gaining weight after ART would be an adverse effect, but these are monkeys that they're gaining weight, but more of like in a monkey model they lose weight because just because of the nature of the model. So they're basically on their trajectory. So I wouldn't want to say gaining weight is going to be a side effect, but our point was that these animals are starting to look healthier than their components that were infected.
So I think we probably need to stop now because we're about 10 minutes over, but this was great to get this much done with two speakers and so many wonderful questions. And again, I want to remind people that the next webinar is on Tuesday, September the 20th, Silencing HIV. Hope MDC site will be presenting that, and the presenter is Pretty Kumar. So thank you so much for.
Thank you very much, Linda.
Bye-bye. Can we possibly get your slides? Is that possible?
Yeah, absolutely. And Linda, one thing I wanted to say is because Jeff Berry is our chair of our CAB. And so, we're working on making education available on this gene editing because we know it's super complicated. So any feedback you can give us or to Jeff and any kind of educational materials that you guys think would be helpful to the community and getting to understand all these things would be absolutely welcomed.
Well, I thought initially that video was very helpful, and I love that red slide with the scissors.
Yeah, I didn't make the video.
The red bow, the red HIV bow, and AIDS bow and the scissor. I loved that, but that was more of a feeling rather than educational, but I thought the video was helpful.
All right. Thank you very much.
And we're always looking for cab members, right, Jeff?
Thank you, Michael and Jeff, and everybody who attended.
Bye-bye.
Really appreciate it. Bye-bye.
Thank you.
Thanks. Bye-bye.